First of all, very good evening. This is Dheeraj Upadhyay from MedGenome, and MedGenome wholeheartedly welcomes you all in this live webinar. Before going further, let me introduce our speaker for today's webinar. So, with us, we have Dr. Priya Kadam, who is the Director of Reproductive Genomics at MedGenome Labs. With so many of years of experience in the biotechnology industry, ma'am have a strong expertise in molecular and cell biology, clinical research, and laboratory management. So, MAM is dedicated to leverage the power of genomics and bioinformatics to provide innovative and personalized solutions for patients and clinicians as well in the field of reproductive health. Today, MAM is going to speak about the significance of low-pass whole genome sequencing and various application and benefits of KaryoSeq. So, over to you, ma'am.
Yeah, thank you, Dheeraj. Welcome you all. Thank you for joining this webinar. And I have been working with MedGenome for the past 9 years. And as Dheeraj mentioned, I take care of the reproductive segment. Yeah, it's my pleasure to welcome all of you. And I'm very delighted to speak about this interesting new test called KaryoSeq. Okay, so this is a low-pass whole genome sequencing test to look for chromosomal abnormalities. Yeah, 1 second. Yeah. So how I'll structure this talk is, we will talk a little bit about the background of CNVs, importance of CNVs, some of the salient features of this particular test, what is the current literature evidence for using low-pass whole genome sequencing for detection of CNVs in clinical setting, what are the indications or clinical scenarios in which you can consider using this test for your patients. We went through an extensive validation to before offering this test to the clinicians. So I will go into a little bit of that. And of course, right now you may be using certain tests for CNV detection, aneuploidy detection. So we will also compare how this test performs in comparison to all other existing tests, and also the test performance in terms of diagnostic yield, a little bit about the lab process, how the sample reports will look, what are the current limitations. And since we have been using this test for about 1.5 months, I will also go a little bit into what has been our early experience with the test. So now, if most of you are obstetricians, gynecologists, fetal medicine people, geneticists, all of you know the importance of CNVs. So we know that these are variations which are found across the genome, and about 4 to 9% of the genome shows this kind of variability. But what is the impact? What we know so far is it may have no impact on the individual. It could lead to morphological variation, adaptability. But what we are most interested in is its contribution to common and rare genetic disorders, and of course, of its role in cancer. Now, it has been known, like copy number variations, as a cause of some of the genetic disorders, and these are losses or gains. And because of this, looking for CNVs is a first-year approach for postnatal evaluation of intellectual disability, developmental delay, autism spectrum disorder, and multiple congenital abnormalities, and also for prenatal evaluation of fetuses with structural abnormalities. So this is quite well known. Now, let us just go back a little bit on the timelines for CNV research. Okay, the circles in green actually tell us the various methodologies which have been used to look for CNVs. And CNVs as a cause of a variation has been known for a very long time, more than 100 years. And in Drosophila melanogaster, the earliest one, and after which in the humans also, when loss of a segment of a chromosome would cause a particular syndromic situation, was known as early as 1963. It was demonstrated then. So now there were various techniques which have been used, and of course we are now coming to the latest technique, which is whole genome sequencing, which is being used for this. Now, CNV as a word has been used for about maybe from 2004, it's about 20 years. And the definition of copy number variations was about more than 100 base pairs a few years ago, which is in 2006, after which the size has reduced to about 50 base pairs. So any variation bigger than that will be considered a CNV. So now, this technique has been gaining wider acceptance. There has been a recent ACMG guidelines, which actually tells the laboratories as well as providers on the various aspects of this, the technical aspects of this, and what we should be looking for when we provide the test, and as providers, what you should be looking for. So, like we said, karyotype, chromosomal microarray, gene panels, exome sequencing, these are the various tests through which a copy number variation can be detected. So what we are focusing on now will be on low-pass whole genome sequencing. So what is low-pass whole genome sequencing? It is an inexpensive, high-throughput DNA sequencing technology which is used to accurately detect genetic variations within the genome. And once the data is available, it will use bioinformatic algorithms and detect variant calls with a high accuracy at very low sequencing depth. So what we mean by sequencing depth, you can look at the image on the right side. So when you do a whole genome sequencing, that is sequence the entire genome, that is done usually at about 30 times depth. You do a whole exome sequencing, it's done about 100 times depth. And there are other panels which are done at a greater depth. So if a sequencing is done anytime, any amount of depth from 0.25 to about 15, actually, the latest guidelines suggest 15. Anything less than 15 times depth is considered low-pass whole genome sequencing. So, but it gives us enough data to be able to pick up the copy number variations. So a little bit more about this particular test. Okay, so we know that it can detect all aneuploidies across all chromosomes and copy number variations greater than 1 MB. If we compare it with qfPCR and FISH, since it looks at all chromosomes, definitely it provides more information. Its resolution is higher than that of karyotype. We have limited the resolution at this point at 1 MB, which is dynamic. We will be going even higher resolution. And karyotype we know can detect over 5 MB. It can be used both for pre and postnatal scenarios, and it is comprehensive. By comprehensive, we mean it interrogates the entire genome, and therefore it is comprehensive. It is affordable. That's one of the major factors of this test, and accurate, and it has been very well validated as well. So it is ideal when investigating for aneuploidies or CNVs at a cost-effective price. So I did reiterate the advantages. We will go over again. Mostly it is cost effective. It is comprehensive because it is genome wide, unlike a microarray, which will have pre-designed probes, obviously very well validated, but this is much more comprehensive than a microarray. It's well validated in our experience, more than 100% concordance for CNVs over 1 MB. You can apply it both for prenatal and postnatal applications, and it has a higher resolution compared to qfPCR karyotyping and a higher diagnostic yield. Higher diagnostic yield has been reported in some papers, and I will show you our preliminary data for what we can see. So these are some of the papers where this technique first came into being in 2016, commercially used as a validated approach. And in certain countries, especially in China, this has been used for a few years now. And this highlighted article is about 3 years experience of using this test and how they have now issued guidelines of how this can be a good alternative test for copy number variation detection. And specifically, there is this particular article which has actually compared. The obvious comparison is with the chromosomal microarray. So what they have found, or this particular paper has found, is it has a slightly higher additional yield, reduced technical repeat rates, less input DNA, which is about 50 nanograms, and also it has increased sensitivity for low-level mosaicism. Closer home, Dr. Shubha Phadke's group has evaluated this technique and come up with this article where they say that there is no net increase. I mean, they did not find the net increase in yield, but the advantage is in that it identifies cryptic CNVs in areas without the probe, because we know that the probes for any kind of array are pre-designed. Better delineation of breakpoints, because it is the genome wide, you could get the exact regions of breakpoint, because even in our validation, we saw the breakpoints would be a little different between what we observe in the low pass whole genome as well as array data. Of course, the technical ease, because we already have the setup for the sequencing and we do huge amounts of sequencing. And of course, the cost per sample. So this is the summary of all the publications for using it in clinics. We say, I mean, in hospitals and for patients, the depth of sequencing can range anywhere from 0.25 to about 5 times. With the increased depth of sequencing, of course, more things can be identified. For now, our data, which is right at the bottom, our depth of sequencing is between 0.7 to 1 time. All sample types have been tested, and sample size of about 138 samples. And this yield is a predicted diagnostic yield. So now, since this technique is gaining wider acceptance and many laboratories worldwide actually have started using it, these are the guidelines which I referred to. They actually give the how the data should be interpreted, what the algorithms should be, and things like that. So coming to the main point, how do you, in what situation would you think of this test or suggest this test? Mostly any high-risk pregnancy where you're looking for aneuploidies, suspecting aneuploidies, high risk on maternal serum screening, or any soft marker where you would think of an aneuploidy. A miscarriage or POC testing. So we see with our preliminary data that this test is preferred. I mean, a lot of clinicians are sending it for testing POCs. For stillbirths, for congenital malformations, any intellectual disability, we can consider doing this test. Suspected microdeletions and duplication syndromes, and high risk, coming back to that. So I'll just go into a little bit into the details of validation, because we went through an extensive validation before offering this test. So what was compared, the CMA data, what was detected on a microarray was compared with the KaryoSeq whole low-pass whole genome sequencing. So there were about 10 normal samples and 138 abnormal samples, and all the prenatal samples of amniotic fluid, chorionis, villus biopsy, product of conception, direct fetal DNA, as well as postnatal samples of peripheral blood was used. What we looked for was whole chromosome abnormalities and any CNV greater than 0.5 MB. So we found 100% concordance for samples with grade of more than 1 MB CNV. So this is the detail of the validation. We had about 8 rounds of it. The first 2 rounds actually were not blinded. So the analysts were actually aware of what they were looking for. But then from validation 3 to 8, it was completely blinded. So they were not aware of what they were looking for, whether the sample was normal or abnormal, and what was the size of the CNV, where it was. And the orthogonal testing, of course, what was with CMA and in some cases NGS. Sample type, you can see the size range was anywhere from 1 MB onwards to whole chromosome aneuploidies. And in each of the cases, we were able to detect the exact copy number variation. So What do the variants look like? This is what it looks like. The first panel is a peripheral blood sample where you can see a 14.2 MB variation. This is a loss over here. Then this amniotic fluid sample actually showed a gain in chromosome 7 and a loss in chromosome 6. The gain was about 25 MB. A 25 MB gain would look like this, and a loss of about 8.3 MB. Sample 3 shows a clear trisomy 18 through of a CVS sample. And in this last panel, there is actually a loss of 1 MB in chromosome 4. So this actually you can't see. That's why I have widened or zoomed in this panel where you can actually see the loss of 1 MB at the bottom. Now, how does this test compare with the existing tests that we have? So we have developed this comparison of the karyotyping with qfPCR with SNP test, SNP microarray. So as you can see, KaryoSeq can detect all aneuploidies and CNVs greater than 1 MB. Of course, SNP microarray, because it uses SNPs at thousands of SNPs and interrogates specifically, you would identify triploidy, uniparental disomy, and absence of heterozygosity. Even low-pass whole genome sequencing will be able to identify these conditions, provided the depth of sequencing is higher, more than 5 times. And that, of course, it will increase the cost. And therefore, at this point, we have restricted it to about 1 time depth, though these incidences will not be found. Of course, qfPCR looks at only 5 chromosomes, and the advantage of karyotype is that it can identify structural abnormalities. Now, the working days is about 8 to 10 for KaryoSeq, and about SNP microarray would be about 10. The resolution is moderate because right now we are offering for CNVs greater than 1 MB. It is extremely cost effective. So maybe that is one of the major factors, cost effective and extremely accurate. So diagnostic yield is for the prenatal samples, but we will go into the preliminary data for comparison. So now I mean, these numbers, I'll just explain these reasons now, these numbers. So how does KaryoSeq compare with Optima, which is the 315K microarray, versus a qfPCR? If you look at pathogenic and likely pathogenic variants, you see it has a comparable diagnostic yield and definitely performs much better than a qfPCR. If we consider the variants of unknown significance, we will see that, I mean, of course, the diagnostic yield, if we include that as a yield, the comparison is similar to that of 315 microarray. Now, why did we use 1 MB as the cutoff? Because we got 100% concordance for that. Now there is research and development underway to reduce this increase the resolution, as in reduce the size of CNVs which we can detect. Now, if you're considering sending a sample for qfPCR, this particular test would be able to detect all that can be detected by a qfPCR. About 99% of what can be detected by Optima, because we haven't included the triploidy here, and about say 83 to 90% of what can be detected through a 750K microarray. So what is the workflow? I mean, this is a lab-related workflow. Any lab person would be interested. So it is pretty straightforward. There would be a sample collection. Any sample type will do. DNA extraction and library preparation. Then we will go for the NGS sequencing and data analysis. We have looked at a few data analytic tools, and even right now the data analysis happens across 2 analysis tools. And the results and interpretation of the report. The report is strictly according to the ACMG ClinChit guidelines for classification of CNVs. So these are the current test codes that we have. These are the 3 main. KaryoSeq without, that is for postnatal samples. Then if you're sending a prenatal sample, an MCC is absolutely essential. and others if a culture is required. There are also combination test codes which have been generated because these are useful. We see that when an exome max is done or a clinical exome is done, doing a low pass whole genome sequencing would give you additional CNV data. So the yield would be higher. So there are various combinations in which this test can be used. So the reports as such is our standard MedGenome report. It would look like a standard MedGenome report. So if a no CNV is detected, of course, it will be in green. If a copy number variation is detected, which is pathogenic or likely pathogenic, it would be in red, and a variant of unknown significance would be in blue. All the details of the CNV as has to be reported according to guidelines will be there in the report. We would have the location, the results, which is the cytoband, the classification, the number of genes which are involved, and the interpretation, which actually will give you some other information on what has been detected. There will be some recommendations as well. So now we should understand what are the current limitations because of how the technique works. Of course, we do not identify polyploidy. Triploidy cannot be identified, but if it is a prenatal sample, an MCC is mandatory or strongly recommended because we can identify triploidy in the MCC itself. So that's why it is currently we cannot identify it by the KaryoSeq. Of course, we can't identify balanced translocations, inversions, monogenic events, or epigenetic events, and uniparental disomies or AOH. And currently, the size limit is 1 MB. And of course, a low-level mosaicisms. Right now, we have the detection limit of about 30%. So this is our preliminary data. So we have been offering this test for about 1.5 months. And as you can see, about 42% of the samples are products of conception, followed by amniotic fluid, which is about 22%. Postnatal samples are about maybe 25, oh, this is more than that, maybe about 30%. So this is how the sample distribution is. And not surprisingly, what we see that KaryoSeq has an yield of about 34% as of now. This is probably because most of the samples are POCs. So we know that POC does have a higher diagnostic yield on a microarray as well. So I've just put the other numbers in comparison. So this is, of course, data over a large period of time, and it includes all sample types. So therefore, this is what is the yield as of now. So I'll just show you very quickly 3 cases which have just come in. So there was this 31-year-old primigravida with increased NT and generalized fetal edema. They did think that it was trisomy 21. The amniotic fluid sample was sent. What it was actually was a trisomy 18. So if you look at this whole genome view at the bottom, you see this very clear gain seen by this KaryoSeq technique. So it is trisomy 18. So it was reported as trisomy 18. The other case was again a 31-year-old woman with a spontaneous miscarriage. A POC sample was sent for testing. And here there were actually two aneuploidies that we found. It is relatively rare. There was a trisomy 22, which can clearly be seen here. Of course, this image should be interpreted like this. This is the copy number state on the y-axis, and these are the chromosomes. So if you come right here, this is trisomy 22. And then here there was monosomy X as well. So the third case is actually a small loss that we identified. It was a 32-year-old woman with a history of 1 neonatal death. At 17 weeks gestation, the sample was sent for testing. So what we found was a 1.5 MB loss in chromosome 17. So here you will not be able to see it that clearly. I wanted to put in a zoom in image, but you can see a small green dot over here. So this is the loss which is identified. So it was about 1.5 MB loss, and the ACMG classification actually classified it as pathogenic. So that's about it. Thank you so much. I'd be happy to take questions.
Hi, so like the floor is open for the questions. If anybody having any query, Dr. Priya would be happy to answer that. You all can raise hand as well. I'll try to unmute them, or like you can just post your question on the chats as requested. Yeah, yeah,
Yeah,
Satish.
it's hello. Am I audible?
Yeah, you're audible, Satish.
Oh
You
yeah,
can
yeah. Okay,
ask the
cool.
question.
So yeah, thanks, Priya. It's a great presentation. So
Thank
my
you.
question is that like, what platform did you use for this all validation? So is it Illumina or MGI? What is the platform? And then
Was
the second question is that let me come. Okay, go ahead. Yeah,
Sorry, your question is what is the platform for sequencing?
yeah, yes,
That's
yes.
the Illumina platform.
Oh, Illumina platform. Okay. So the second question is that is there any difference in between diagnostic yield in the context of the sample type?
Sorry, any? What about the sample type?
The difference in the diagnostic yield.
Diagnostic yield? Oh
In
yes, we do have. Like I did show, there was a difference, but that is a standard thing which we expect actually.
Okay, so there
Yeah,
are no surprises, you mean?
no surprises. Yes, because the POC we see maximum diagnostic yield, and the others it will be relatively lower.
So you got any chance to look into Y chromosome microdeletions by any chance?
By chromosome deletion, if the size is greater than 1 MB, we should be able to pick it.
Okay, so okay, thank you.
Thank you.
Okay, yeah, Abdul, I have unmuted you as well, so you can just park your code. Ask the question.
Hi, this is
Hi,
Dr. Abdul here. Hi, Dr. Priya.
good evening.
Yeah, good evening. I just have one question. How we can assure when we sequence like 1x? Because if you see IGV, it will be just 1x, and how we can assure that this is true positive result?
That is the validation actually. With the validation, we have been able to do it. And the algorithms are imputation algorithms. So there are two different algorithms which have been used, which, we haven't had a false negative so far. I mean, with any technique, there is a slight possibility of that. But so far, it has been a well-recognized technique. We do it at a depth of 0.7 to 1, sometimes even higher if a sample fails the QCs, right? So we will do it at a higher depth. There have been publications which have sequenced at even 0.25.
Okay,
Yeah.
okay. So
So
you
the
give
higher
some
the
in raw
higher.
data.
Yeah, please go ahead.
In raw data, do you give any cutoff for probe binding? How much percentage of probe binding
Yes.
should not be or should be there? Something like that.
Yes, we do have the QC matrices. So the QCs have to pass.
Okay.
That
Okay.
takes
Thank
care
you.
of it. Yeah.
Thanks.
Yes, Adnan, you can ask your query. Adnan, you are unmuted. Yeah, funny. You are unmuted, funny.
Yeah. Hi, Dr. Priya. A great presentation and a great work. So, so I just want to understand the algorithms because copy number identification in whole genome is very tricky, and there are no guidelines as such which sort of suggest how do we go about it. So, is this an algorithm which is proprietary built, or is this something? Is there any recommendations available to understand the identification, how true it is, or how close we are to the true positives?
Right. So unfortunately, at this time, I'm not at a liberty to tell you about the algorithms. We do have an in-house one as well, but we also have two commercial ones, right? So, of course, it can be done offline also. So these are well-recognized algorithms, and they are validated as well. So for any technique, it is validation against an orthogonal test, right? So as of now, we do see a highest level of concordance.
Okay. And how do you sort of achieve a uniform coverage of your sequencing at such a low depth across the whole genome, considering that the sequencing at centromere and sequencing at the terminal end of the chromosomes are very difficult?
Right.
At this depth, I mean, how does this been achieved?
What has been achieved, I mean, so far, what we can see, I mean, that is a difficult question. I don't really know exactly, because that goes completely into the sequencing part, the laboratory based. Maybe some of my colleagues will be able to answer that. But definitely it is I mean, what we see is these are imputation algorithms is what we have understood, in that they kind of generate the data. I mean, I think this is something which we should take.
Yeah,
I wouldn't be able to. Yeah.
I want to add, just ask one question, Dr. Priya.
Yeah.
You have done orthogonal validation, but
yes,
if you are comparing between Illumina and with KaryoTeq or qfPCR, it's better to compare with the same technology with the intralab validation rather than why? Because you are sequencing here 0.5x or 1x, and you're comparing with qfPCR, which is
no, we are comparing with the microarray. The orthogonal testing was microarray. Yeah.
I done the intralab validation with doing some whole same WGA.
That is actually underway. And as we do all the PT testing and intralab validation, that is underway.
Okay. But there's some very few labs doing.
Yeah, that is another this thing. Another.
Myanmar and some other labs doing.
Yeah.
Thank you.
Thank you.
Yes, Soumya, you can ask your query.
Good evening, ma'am. Actually, this is not much of a query. I just wanted to, because I was going through your presentation, I just wanted to know, like you mentioned that Lupa's sequencing technique that is KaryoSeq, can detect up to 99% of what qfPCR can detect. I just missed that 1%. What is the 1% that it cannot detect, ma'am?
Triploidy.
Triploidy. But in one of your cases, you had shown that triploidy, okay. So it cannot detect triploidy.
Yeah. What that particular slide showed, whatever can be detected on qfPCR can be detected by this technique.
Okay.
Everything, because all the aneuploidies of the 5 chromosomes or even the 22 all other chromosomes, it can be identified. But when you compare it with 315 optima, okay. Then we have given about 1%, it won't be able to detect, which is the triploidy. So even triploidy, actually we have had a recent case where there is a method of actually analyzing it where we suspect it, we can suspect it. So that is the thing. And because it could even look like maternal cell contamination, right? Triploidy looks like that as well. So those will not be, but the way I think we can kind of not have a clinical impact is that any prenatal sample will have to go through an MCC. So we know that those markers will be able to identify a triploidy.
Yeah, because qfPCR has a built-in MCC check also.
Yes.
So does this technique use a different MCC kit or something like that before going for the KaryoSeq?
We recommend an MCC, an actual regular MCC. Yeah.
Okay.
Yeah. And one other point I wanted to tell you, if the depth of sequencing increases, right, we go to about 5 times, we can actually identify triploidy as well. And anything that can be identified SNP based, like
because in the starting of the presentation, you mentioned that it can go up to 15x.
That is the guideline, actually, the 2023 guidelines. Anything less than 15 depth is considered a low pass. That's just a definition, but all the publications which I've found so far, because the Alka Chaubey paper is 5 times, then the whole genome sequencing which has been done, I mean, the first whole genome sequencing was done about 7.2 times. Yeah,
Okay.
yeah.
So we are doing it for, like, we do it 1x and we find something, then we can do it again. So
Yes.
we can just increase the number of runs and increase the depth also.
If we are
Yeah.
not sure about what we are seeing. Yeah.
Okay. Yeah. Thank you, ma'am.
Thank you.
Any other questions? If not, then thank you once again, ma'am, for this wonderful session. And I hope like the audience got benefited with your talk, and the same time, like about the low pass significance of the low pass whole genome sequencing and how the KaryoSeq would be changing the paradigms about it, ma'am.
Yes, thank you. And thank you for the questions as well. And you could actually connect with us, the tech support, and we can try to resolve whatever queries you have.
Sure, ma'am. Sure, ma'am. So thank you so much once again for attending today's webinar, and I hope like in coming days again, like we'll be having some more webinars similar to this various topics. Thank
Thank
you
you.
all.
Yeah, thank you so much,
Thank you,
Deepak,
ma'am.
for organizing, and thank you all for attending.